Hello, everyone. Oh, okay. Hello, everybody. Can you hear me?
Hello, yeah, we can hear
Oh,
you.
good.
Yeah, hi, Professor. I can hear you. I just want to ask, is this recording, is it being recorded?
Yes,
Okay.
all classes will
When will
be
you get a
recorded.
chance to upload it?
Sorry,
When will you get a chance to upload it?
tomorrow morning. Tomorrow,
Oh,
because I don't
okay.
record myself. I have to wait for
All right.
the tactic support to convert it to YouTube and send to me. Okay. Okay, so let's start class. So today we have a lot to cover, but I should say first that I was really pleased with how you all, most of you, were able to access Exam Soft and to take the mock exam. If you have questions about this process, you can ask them to me after class. I will stay on and then you can ask me questions. Okay, so in the last class, we spent a lot of time talking about the plasma membrane, its properties, and how it forms an outstanding barrier to not only a barrier to prevent microorganisms to invade the cell, but also to manipulate and to selectively allow certain molecules and ions to get into the cell in a highly selective manner. Okay, so now the next question, what is in the cell? And what are the function of all these organelles and structures? So we'll start with the cytoskeleton. So you think about the cytoskeleton in a cell like the cytoskeleton in our body. So the cytoskeleton, cyto means cell. Every time you see that prefix cyto, it means cell. So the cytoskeleton allows the cell to have a specific shape and supports the cell. It also, so you don't want in the cell all the organelles and all the different structures to be just mixed up and floating around. Everything has its own place, and so the cytoskeleton also helps organize all these structures that are in the cell and provides for movement. So in the cell, there are vesicles and, for example, mitochondria that move around from one area to the other. And that movement needs a highway, just like cars need a highway to move from one area to the other. In the cell, we also have these highways that are made up of cytoskeleton proteins. And the cytoskeleton is very active part of the cell, and it's not stable. So it can come apart and then organize itself. So it's very dynamic. Okay, so what makes up the cytoskeleton? So our skeleton is made up of bones, and we have bones of all shapes and all different sizes. So the cytoskeleton of the cell has what we call filaments. So these filaments are made up of proteins. And there are different types of filaments. The ones that are thinnest, they're made up of this protein known actin, which is a highly abundant protein in the cell. And it makes up the microfilaments. As the name indicates, that means they are the thinnest. And for example, they support the microvilli. These cells are present, for example, in the small intestine. And the microvilli are facing the lumen of the small intestine. And this allows the microvilli allow for an increase in the surface so that the absorption can be more efficient. Absorption of nutrients and so on that are inside the lumen of the small intestine. So by having these microvilli, you increase the surface that allows these nutrients to come into these cells. So these microvilli, they are supported, their shape and so on is supported by actin. Then intermediate filaments. So that means that the diameter of these filaments is a little bit bigger than the microfilaments. And the intermediate filaments are made up of different types of proteins, like vimentin. And their function is to anchor the organelles. So as I said, you don't want the mitochondria and lysosomes and so on to be chaotically placed in the cell. So you want them to be anchored to certain filaments and so on. And then we have the thickest of all filaments, which are the microtubules. And these microtubules are made up of tubulin. This protein tubulin is small, you see here. These are, each of these circles represents a tubulin. And they what we call polymerize. So they make up these highways. So they bind to each other and they make these long structures. I didn't cough the whole day. I promise you. So these make up the highways for vesicles and organelles to move from one area of the cell to the other. They're also present in flagella, cilia, and centrosomes, which we will discuss soon. So in these microtubules, you see here, you have the light green and the darker green. Each of these is one molecule of tubulin. And they come apart, they disorganize and polymerize, forming these long highways. So the long highways have specific motors that carry the organelles and vesicles along. So we have kinesin and dynein. So those are the motors that carry the clients in opposite directions. So kinesin carries their cargoes from the center of the cell to the periphery. So we say that it's anterograde. So from the center of the cell to the periphery of the cell. So the center would be the nuclear location, and then will carry these cargoes towards the plasma membrane, while dynin will carry the cargo from the periphery towards the center of the cell. So you see here one of these proteins like kinesin carrying a big vesicle. And of course, this movement along the microfilaments that are made up of tubulin is ATP dependent. So the cytoskeleton is just a transport highway for the cell. Well, that's the microtubules, because the different components or types of cytoskeleton that have different functions. So you see here, I'm going to show you, I hope this works. Let's see. Yeah. So you see
redness and bumps. Get irritated. Redness and bumps. Get irritation from the pubic hair.
this is not good. Oh, come on. Okay. Now here you see the tubulin molecules arranged in these microtubules, and this could be kinesin. So you see that the protein kinesin, how it is functionally and structurally, its structure, the protein has different primary, secondary, and tertiary structure, and how it moves along the tubulin, the microtubules. And of course, you need a lot of ATP. Okay, now we move on to the centrosomes and centrioles. So what are these structures? The centrosomes and centrioles, they're involved in the organization of these microtubules. So this whole structure here in green represents a centrosome. So centrosome is a highly dense area in the cytoplasm that houses the centrioles. So the centrioles are these cylinders, there's two of them in each centrosome. The centrosome is the site where the microtubules are organized. These black structures here, lines, represent the microtubules, and they have a negative side, which faces the centrosome, and the positive side that faces the opposite. So these, the centrosome with its centrioles is important for organizing the microtubules and also for the mitotic spindle during mitosis. Remember that the mitotic spindle is involved in the movement of the chromosomes to different poles of the cell. So let's see if this works. So you see here a cell going through mitosis, and you see the microtube, the mitotic filament. You see the mitotic spindle moving, the chromosomes are in red. So you can see this mitotic cell being organized and dividing into two cells. It's a beautiful process.
I can't see the video.
Oh, you guys can't see the video? No one?
No.
No. Okay. Let me see. Stop sharing. Now if I share here. Now you can see it, right? Can you see?
Yeah.
Yes. So you see the green represents the mitotic spindle and the red is the chromosomes. And you see the cell, the chromatin is organized into chromosomes, And then the spindle pulls them apart so that one cell is going to be divided through mitosis into two cells. But you see here that the centrosome and centrioles are responsible for organizing this, this mitotic spindle. Okay. Oh, now I have to go back to doing this and doing this. Okay. Very good. Okay, so what is interesting is that here you see these two structures or tubules are called centrioles. And now if we focus and magnify, what do these centrioles look like? So the centrosome is this whole area that contains a dense cytoplasm and then the centrioles. So if you focus on the centrioles, they're made up of microtubules. Remember tubulin, each of these, the microtubules, there's 9 of them, and they are organized in groups of 3, none in the middle. So you have 9 groups of 3 microtubules each. This is how the centrioles are organized. And you see here how the centrosome, this whole area that contains 2 centrioles, is organized. And then here you see the centrioles that are organizing the mitotic spindle that allows the chromosomes to be pulled apart to each pole. Okay, how about cilia and flagella? They also are made up of microtubules. Remember, when we talk about microtubules, these are the thickest of the filaments and they're made up of tubulin. So the organization here of these microtubules is different from the centrioles. Here in the microtubules, the tubulin microtubule, there's 9 groups, but instead of being organized in triplets, they're organized in 2 doublets. So there's 9 groups, each of each unit having 2 microtubules. And the other difference is that cilia and flagella are protected by the plasma membrane. So for the centrioles, there's no plasma membrane protecting them. But for the cilia and flagella, there's a plasma membrane that surrounds the cilia and flagella. So both cilia and flagella are organized in 9 plus 2. What does that mean? That means that there's 9 groups of microtubule and 2 in the middle. Okay, so here you see the flagella and cilia. What is the difference between flagella and cilia? So flagella, there's only one. You see here a beautiful sperm. So the flagella, remember, this sperm tail is made up of a structure like this that has 9 microtubules and 9 pairs of microtubules and 2 in the middle, and it's surrounded by a plasma membrane. And this is used for the swimming of the sperm, or flagella in some other structure. Now here you see the cilia. There are many of them. So flagella is just one, and cilia, there are many of them. Now, where are the cilia? The cilia are located in many different parts of the cell, and they are highly abundant in the respiratory tract. Why? Because they trap particles, and then to get rid of these particles so that they don't go into the lungs, someone will cough, the individual will cough and remove those particles. So the cilia are moving in waves to allow the individual to get rid of those particles that might be accumulating in the respiratory tract. Now, why do individuals that smoke sometimes cough much more than a non-smoker? Well, what happens is that the nicotine inhibits this movement, this wave-like movement of the cilia that is important to remove these minor particles that can come through the trachea. So if the cilia are paralyzed by nicotine, then the individual has to cough in a much stronger way to get rid of these particles. So that's one of the reasons why nicotine is damaging, is that it induces this high, frequent, and strong coughing of the individuals because the cilia are paralyzed. Okay, now we move on to the ribosomes. So the ribosomes are the organelles in the cell that are responsible for protein synthesis. And you know that the protein synthesis, the synthesis of one protein, depends on one gene. So there's not a one-to-one relationship in humans, between a gene and one protein, because in humans, one gene can give rise or can be transcribed, translated into more than one different type of proteins. And I will explain that soon. But let's go back to the ribosomes. So the ribosomes, some are free, as you can see here. They're not attached to any organelle. And they form these groups that we call polyribosomes. Poly means a group, more than one. So you see here in red, each of these circles represents one ribosome. And if they're not attached to the ER, to the endoplasmic reticulum, they're free in the cell. And proteins that are synthesized by these ribosomes, they are targeted, they remain in the cytoplasm. But there are other ribosomes that are attached to the endoplasmic reticulum. You see here this cisterna, these tube-like structures that form the endoplasmic reticulum. And you see the gray, the black dots represent ribosomes. And they are attached to the endoplasmic reticulum. And what is their function? So these ribosomes that are attached to these tube-like structures that form the endoplasmic reticulum, they form proteins that go into the endoplasmic reticulum, and then they are transported via the Golgi to membranes, such as the plasma membrane or the lysosome, and so on. So here, the ribosomes that are free are known as polyribosomes. So you see the ribosomes that are attached to the ER, they're going to end up on the plasma membrane or on the lysosomal membrane. So the proteins are synthesized and they get into the ER. From the ER, they're transported to the Golgi. And from the Golgi, they go on to their location in the plasma membrane or in the lysosome. However, the ones that are free ribosomes, the proteins that are synthesized by free ribosomes, they stay in the cytoplasm. So the ribosomes contain 60% ribosomal RNA that is synthesized and made, is assembled in the nucleolus, and 40% of protein. So these ribosomes are assembled in the nucleolus, and I will explain what the nucleolus is. So here you see a large subunit and a small subunit of the ribosome. And what are their functions of these two subunits? So the small subunit that is represented in lavender is the subunit that binds to the messenger RNA. So the messenger RNA is a nucleic acid that is synthesized in the nucleus. After it's synthesized in the nucleus, it moves out to the cytoplasm and it binds to the messenger RNA of the ribosome. Now, this large subunit is closely assembled to the small subunit. And what is its function? Its function is very important. Its function is to make a peptide bond. So you have this large and complex organelle, the ribosome, and its function is to form a peptide bond. And if you remember what is a peptide bond, A peptide bond is a bond, a covalent bond that links together two amino acids. So once the amino acids are linked by a peptide bond, now they're called residues. And remember, this is a dehydration synthesis. So when you connect two amino acids to form a dipeptide, so they're connected by a peptide bond, then a molecule of water is released. Okay, so here you see the large ribosomal subunit, and you see these sort of elliptic structures. So these are very important because there's another RNA that's called transfer RNA, and it brings the amino acids to the specific sites on the ribosome. So then you have two transfer RNAs, one here, with their amino acids sticking out, and then the ribosome is able to connect those amino acids by a peptide bond. So all this happens in the large subunit of the ribosome. So it depends on so here you have the messenger RNA that is synthesized in the nucleus and binds to the small subunit. And these two sites here, they're the sites for the transfer RNA, which we will talk about later. And the transfer RNA, as its name indicates, transfer. Each transfer RNA is connected to a specific amino acid. So you have one transfer RNA with its amino acids sticking out, another transfer RNA with its amino acids sticking out. And when they come to the large ribosome subunit, because they're so close together, there's a peptide bond that is formed. Okay, now let's move to the ER. The ER is the endoplasmic reticulum, and it's made up from these tubules that we call cisterna. And there's two types of ER, the rough ER and the smooth ER. The rough ER I already talked about, and it's the one where the ribosomes are bound to. So like here, right? This is an electron micrograph showing the rough ER. So the rough ER, you see here, the nucleus with a nuclear envelope. And rough ER, these cisterna, these tubules are very close to the nuclear membrane. So you have lots of ribosomes attached to this rough ER, and they are the sites of protein synthesis. And these proteins are going to end up in the plasma membrane or the lysosomal membrane. So the ER is the site of protein synthesis, but also phospholipids and carbohydrate chain assembly. So remember, there are some proteins that are what we call glycosylated. That means these proteins have sugars, carbohydrates attached to them. And this is where, in the ER, this is where they are assembled. Now, the smooth ER that's represented here, there's no protein synthesis here because there's no ribosomes attached to the smooth ER. So what is its function? The function is to synthesize lipids, cholesterol, lipoproteins, phospholipids, steroid hormones. These are all lipids. Where are they synthesized? In the smooth ER. But then another very, very important function of the smooth ER is that it stores calcium. And calcium is very important for muscle contraction. It's actually critical. So the ER stores this calcium, and when there's a signal coming from a neuron, the signal is transmitted to the small ER, and calcium is released to induce muscle contraction. So the smooth ER is important. Why? Because it's the site of lipid, the major site of lipid synthesis, all these different types of lipids, but it's also a site where calcium in the cell is stored. So calcium is mostly stored in the ER. Okay, so the Golgi, I've been talking about the Golgi. So what you see here in pink, this is a nucleus, This is the endoplasmic reticulum. I would say this is a rough ER. And then here's the Golgi. So the proteins that are going to end up in the plasma membrane, on the membrane of the lysosome, they are synthesized in the rough ER. But then how do they get to their sites? How do they get to the plasma membrane? So the Golgi is involved in bringing, in targeting those proteins that are synthesized in the ER to, and bringing them to the plasma membrane. So the Golgi is formed of also cisterna, and we call the cis Golgi, then the mid Golgi, and the trans Golgi. So there's these three different types of Golgi. The cis Golgi is the closest to the ER, and the trans Golgi is the closest to the plasma membrane. So how does this happen? So the proteins are synthesized in the ER, then there are vesicles that bud off and fuse with the cis the cis Golgi. And then in the cis Golgi, these proteins can be decorated also with carbohydrates, which serve as the zip code to where this protein is going to end up. So certain carbohydrates, such as mannose. Mannose is a carbohydrate, a sugar, that is attached to certain proteins. When the protein is decorated by this sugar, mannose, that means it's going to end up in the lysosome. But other proteins that are synthesized in the ER, they are decorated by other carbohydrates. So they're going to end up, they go through cis, mid, and trans Golgi, and they're going to end up in the plasma membrane. So all these channels that we've been talking about when we talked about the plasma membrane, that there are all these channels that are proteins, they go through this process. They are synthesized in the rough ER, then they go through the Golgi, and then they end up at the plasma membrane. So you see here what happens. So the ER, you see now here the ER with ribosomes, and the vesicles that are come off the ER, and they fuse with the cis Golgi, and then they can be decorated with carbohydrates and so on. And then there are vesicles that are bud off the trans Golgi, and they can be used to form secretory vesicles, or for proteins to be embedded in the plasma membrane, or other proteins are transported to the lysosome. So this transport, this complex transport from the ER to the Golgi, and then locating the protein in their proper location at the plasma membrane. There's many factors that can contribute for this transport to not happen. So these proteins have to be decorated with carbohydrates and so on. But if the proteins are mutated, that means that they will not be transported and they will not be present at the plasma membrane. So there's one disease known as cystic fibrosis. And the problem with cystic fibrosis is that there are mutations in this protein that's a chloride pump. So the chloride pump pumps chloride ions from inside the cell to the outside. So if it's not properly transported to the plasma membrane, then it's enriched in the cell, and the cell will have high concentrations of chloride ion. And these high concentrations then dehydrate the cell and dehydrate the mucus that lines the respiratory tract. So this mucus now becomes very, very thick. And because it's at 37 degrees and it's highly viscous and thick, it's an optimal environment for bacteria to grow. So what happens is that the respiratory of these individuals that suffer from cystic fibrosis, the lumen of the respiratory tract becomes thinner and thinner, and they have a lot of problems breathing. Sometimes they have to be connected to a respiration machine to help them. So here, up to a few decades ago, these individuals would die at the age of 10 because they couldn't breathe properly and they had all kinds of complications. Now there are ways to improve their living, and they can live to about 40 or more years of age. And one strategy is to give them antibiotics. Why? Because I just told you that there's a lot of bacteria that grow in this mucus that lines the respiratory tract. So if you give them specific antibiotics, those bacteria will die and this mucus will become thinner and it will help the patient breathe. So what is showing here is this chloride pump. So when it's placed properly at the plasma membrane, then the individual has no problem and the mucus is not so viscous. But if the the chloride pump is not located properly at the plasma membrane, then you see how here the mucus is represented very thick and allows for bacteria to grow. So question, what about the proteins made in the free ribosomes? Where do they go? So those are the polyribosomes. The proteins made by those ribosomes, they remain in the cytoplasm because there's many proteins in the cytoplasm. There's some proteins that are at the plasma membrane, but there are many proteins that are located in the cytoplasm. So those proteins are synthesized by the free ribosomes.
Professor, I have a question.
Yes, please.
I was also wondering, so back in the other slide, is it that there's too many chloride ions within the cell or outside of the cell?
So what happens is that this pump pumps the chloride ions out. So if it's not functioning properly, there's a lot of chloride ions that stay in the cell. And because they stay in the cell, then the viscosity of the mucus is highly increased.
Okay, thank you.
Okay. Okay, so now we move to the lysosome. So here you see the Golgi, and you see that the proteins that are made come from the ER. And these proteins that are going to be targeted to the lysosome, they are targeted, they are decorated by a specific carbohydrate that is called mannose. And then there are receptors specific for this carbohydrate, mannose, that will deliver these proteins to the lysosome. I think it's that it's too much mucus, which makes it very hard to pump out chloride ions. Now, the problem is it's the opposite. It's that the chloride pump that is not working properly that leads to the thickening of the mucus. Okay. Okay, so here we talked a lot about the lysosomes as being the stomach of the cell and the lumen of the lysosomes. So the lysosomes have a phospholipid bilayer and the lumen is highly acidic. So what does it mean if it's highly acidic? That means that it has a high concentration of protons, H+, that's a proton. So how is it possible that inside the lysosome, the concentration of protons is much higher than its outside environment? So the pH in the lysosome is around 5. So what happens is that on the membrane of the lysosome, there's this pump. It's a hydrogen pump that carries protons from low concentration to high concentration. So this is active transport that requires ATP. So that means that the hydrogen ions go from low concentration to high concentration. So they are moved by active transport that is ATP dependent. So a high concentration of hydrogen ions inside the lysosome leads to a decrease in pH so that it's acidic. The lysosomes have specific receptors to recognize the specific carbohydrates on the proteins? Yes. So the receptors move along the Golgi and so on, and then they're going to end up in the lysosome. So inside the lysosome, there's many, many enzymes. We call them acid hydrolases. Hydrolases, so they use water to break apart macromolecules. So we talked about all these macromolecules, proteins, carbohydrates, lipids, and so on. So these enzymes are known as nucleases. Nucleases break down nucleic acids into their monomers. Proteases break down proteins. Glycosidases hydrolyze carbohydrates, and so on and so forth. So there's 60 or more different types of acid hydrolases that are kept enclosed in the lysosome. And the lysosomes are not all the same. Some might have more of this type of proteases. Another one might have more of this type of lipases. So here you see an image of this hydrogen pump. So some of these acid hydrolases might be mutated. And if they're mutated, they can lead to disease. There are many, many diseases that are associated with mutations in these hydrolases that are present in the lysosome. Just to give you two examples, Tay-Sachs disease. These two diseases are preponderant in the Jewish population. So Tay-Sachs disease is caused by mutation in this enzyme, hexoaminidase A. And what it does, because it's mutated, there are glycolipids, this type of glycolipids that are broken down by this enzyme, they accumulate in nerve cells. And this leads to problems with brain degeneration, blindness, paralysis, and the children die at around 5 years of age. So it's a major effect due to the mutation in this specific lysosomal enzyme. Another disease that is also associated with a mutation in one of these lysosomal enzymes, this one is called glucocerebrosidase. And this glucocerebrosidase is abundant in macrophages. So if it's mutated, then its target, the glucocerebroside, is not broken down and accumulates in these macrophages. So these red structures here, they're red blood cells, and here you have an enormous macrophage. So these enzymes that are enriched in the lysosomes of macrophages, if they don't have this enzyme, the enzyme is mutated, then there's glucocerebrosidase that accumulates, and this leads to Gaucher's disease. If it presents itself in the infantile age, it can lead to nervous system degeneration and death. If it presents itself in juvenile, because there are mutations that can occur spontaneously, they lead to anemia and large spleen. So these are two examples of two enzymes of two diseases that are associated with mutations in lysosomal enzymes. So there's more than 60 different types of these enzymes. Okay, now peroxisomes. These are another type of organelles. They also have a phospholipid bilayer. And what is the major enzyme present in peroxisomes? The major enzyme breaks down is catalase. What does catalase do? Catalase is an enzyme that is dedicated to breaking down hydrogen peroxide. You know that hydrogen peroxide is very toxic. So when we have a wound, we disinfect it sometimes with hydrogen peroxide. Why? Because it's toxic to bacteria and microorganisms that might be in that infected area. So but our own cells produce hydrogen peroxide. So they produce hydrogen peroxide in the mitochondria. So we need to get rid of it. So this enzyme that is located in peroxisomes, the enzyme is known as catalase, breaks down hydrogen peroxide into water and oxygen. So you see here one organelle, the function of which is dedicated to breaking down hydrogen peroxide. It also has other functions such as detoxifying alcohol, fatty acids, and so on. Okay, now we go to mitochondria. So could we also say peroxidase is a major enzyme? Yes, but here we are calling peroxisomes. So peroxidase would be catalase. Okay. So catalase destroys hydrogen peroxide, correct? Okay, now mitochondria, this is very, very interesting organelle, and we think it originated from the symbiosis or the joining of a cell with a bacteria. So why? Because it has its own DNA. It has two phospholipid bilayers. So an outer membrane and an inner membrane. The outer membrane is a phospholipid bilayer, and the inner membrane is a phospholipid bilayer. So the inner membrane, you see that it has these folds. And what is the reason for it having these folds? Because it has many, many enzymes that are used to make ATP from glucose. You see here one of these folds, and the little red dots represent enzymes. And through the mitochondria, the breakdown or the production of ATP is known as the Krebs cycle. And it's a very, very complex type of mechanism to go through many, many enzymatic steps. But it's very efficient because one glucose that goes through the mitochondrial cycle can give rise to 36 or 38 ATPs. So it's a very, very efficient machinery to produce ATP. Now, mitochondria can self-replicate. That means that one mitochondria can give rise to many mitochondria and so on. And inside the mitochondria, there's a mitochondrial DNA. And this mitochondrial DNA is different. It's a different molecule from the nuclear DNA. The nuclear DNA encodes from has many, many genes, such as 25,000 genes. Now, the mitochondrial DNA has only 37 genes that encode proteins that are kept in the mitochondria. Now, what is interesting about this DNA, mitochondrial DNA, is that it's inherited only from the mother. So this is very important, and this is how important for going through and deciphering the inheritance of an individual. So it's not the nuclear DNA that is used, because the nuclear DNA comes from the mother and the father, but the mitochondrial DNA comes mostly from the mother, because mitochondria in the sperm are present mostly in the tail, and the tail is lost once the sperm head gets into the egg. Okay, so here this drawing just shows how complex, but how efficient the cycle that goes on in the mitochondria is to produce ATP. So glucose gets into the cell and goes through a process known as glycolysis to produce ATP. So the production of ATP in the cytoplasm is not so efficient. So from one molecule of glucose, you get 2 ATP. But if glucose goes through chemical reaction and it's converted to pyruvic acid, now pyruvic acid gets into the mitochondria, and going through the Krebs cycle, then it produces 36 to 38 ATP. So you can see the difference. Within the cytoplasm, glycolysis gives rise to 2 ATP from one molecule of glucose. But if it goes through the mitochondria, then one molecule of glucose gives rise to 36 and 38. So what is the factor that allows glucose to go through this Krebs cycle? So this is oxygen dependent. So if you don't have enough oxygen, this Krebs cycle cannot occur. So you have a very low production of ATP. So the Krebs cycle, the electron transport is an oxidative phosphorylation, is highly dependent on oxygen. And here is where we have oxygen peroxide being produced. It's through these reactions in the mitochondria. Okay, so here one hypothesis is that we all are now present on Earth. We all came from one woman known as the mitochondrial Eva. Now, how do you get to that? So studying the mutations that occur in the mitochondrial DNA, because it's only transmitted from female to female. So this mitochondrial Eve lived 200,000 years ago, and she lived in Africa. So it means that we all came from this female that lived in Africa in 200,000 years ago. And how is this studied? By looking at the mutations in the mitochondrial DNA, because it's only inherited from female to female. Okay, now let's move on to the nucleus. So the nucleus is a very, very large organelle. It's the biggest organelle in the cell. And its function is like the skull. The skull protects the brain. Well, the nucleus protects the DNA. The DNA never leaves the nucleus. I should say the nuclear DNA, not the mitochondrial DNA. So the nuclear DNA never leaves the nucleus. So the nucleus is there to store this DNA. This storage is very important because it prevents most of the mutations to occur, because it's protected, it's encased in this organelle that's the nucleus. So most cells contain one nucleus per cell. But for example, the red blood cells, when they are mature, so they go through different steps of maturation, when they are mature, they lose their nucleus and they become just a storage for hemoglobin. But there are also other cells, the skeletal muscle cells, they contain more than one nucleus. Is there any condition that causes a deformed nucleus and the DNA leaves the cell? Not that I know of. There might be, but at this point, I don't know of. I don't think the cell could survive if the DNA would move out to the nucleus. Because then how would it go through the process of transcription and all this? So I don't think the cell could survive without an efficient nucleus to protect the DNA. So now let's look at this cell. So the blue represents the nucleus. So it contains more than one nucleus. So these are the skeletal cells, skeletal muscle cells. So these cells need a lot of ATP, and they go through a lot of damage because you have these proteins that allow for the movement of the muscle cell, the stretching and going back to the original step. So there's a lot of protein damage. So that means they require a lot of transcription and translation. So that means they require a lot of protein synthesis. So they developed in a way that each cell, this is one skeletal muscle cell, has more than one nucleus. And the advantage of this is the ability to produce more proteins, synthesize new proteins, because there's a lot of wear and tear going on in these muscle cells. So here you see represented a nucleus with a membrane. So the membrane has two phospholipid layers, bilayers, just like the mitochondria. So the mitochondria had an outer membrane and an inner membrane. Each of the membranes were a phospholipid bilayer. The same in the nucleus. The nucleus has two phospholipid bilayers. So it has two membranes, an outside membrane and an inner membrane. And the fluid that fills the nucleus is known as nucleoplasm. And you see here a nucleolus. Most cells have only a nucleolus, and this is where the ribosome assembly occurs. And here is where the DNA is stored. So when the cell is not dividing, the DNA is dispersed, and we call it chromatin. When the cell is going through mitosis, then the chromatin comes, densifies, and you can notice, you can see the chromosomes. Okay, one important thing is here, remember that the nucleus has to allow the messenger RNA to move out, and there's many molecules that go in and out across this nuclear membrane. So the nuclear membrane has these large pores. And these large pores allow for macromolecules, such as the messenger RNA, to go out. So what regulates what can cross this nuclear membrane? So these pores are highly, highly complex. You see here a nuclear pore, and you see all these proteins that form the nuclear pore, and they are very complex and highly regulated, allowing for macromolecules to go in and out. So the space between the two nuclear membranes is known as the perinuclear space. So the nuclear membrane with these two phospholipid bilayers has these large nuclear pores that allow for macromolecules to go in and out, but in a highly regulated manner. It's not like the plasma membrane. The transport in and out of the nucleus depends on the regulation by the proteins that make up the nuclear pore. Okay, so in the chromosome, that's where the DNA is located. So in humans, we have 46 chromosomes, or let's say 23 pairs. In somatic cells, that means in all cells of our body, except in the sperm and oocyte. These cells only have half of the number of chromosomes. So somatic cells, which are the cells throughout the body, they contain 23 pairs. But in the sperm and oocyte, there's only 23 chromosomes. And this makes sense, because when you have the sperm and the oocyte coming together, then each of them contributes with 23 chromosomes, so you end up with 46 or 23 pairs. So out of these 23 chromosomes that are present in the cells, 22 are known as autosomes, 22 pairs are known as autosomes, and one pair is known as a sex chromosome, because one is present in the sperm and the other one is present in the oocyte. And that's what we mean by X and Y chromosome. Okay, so what are the functions of the nucleus? So the nucleus protects the DNA, but what happens inside the nucleus? Inside the nucleus, you have a process known as replication. So what is replication? Replication is the process by which a DNA molecule is replicated, is divided, or produces two DNA molecules. And why is that necessary? Why is it that one DNA molecule needs to give rise to two DNA molecules? Well, if you think about mitosis, right? Mitosis is you have from one cell, you have two cells. So each of those cells has to have the complete DNA molecule. So that's why you need to replicate the DNA molecule when the cell is dividing through mitosis or meiosis. So that process is known as DNA replication, when one DNA molecule gives rise to two DNA molecules. In the nucleus, you also have a process known as transcription. This is the first step that occurs when a protein is synthesized. So what does transcription involve? Transcription involves the synthesis of a messenger RNA molecule from one strand of DNA. So this messenger RNA is important for protein synthesis. It's different from DNA replication. DNA replication, you get from one DNA molecule, you have two DNA molecules being produced. In transcription, you have one DNA molecule giving rise to one messenger RNA molecule. That's the process of transcription. Now, the DNA molecule is huge. Its size, if we would stretch it out, it will be 6 feet. So you need to fold and fold and fold that DNA molecule to pack it into the nucleus. So here you see the two double strands with the base pairs sticking out, and they're linked by hydrogen bonds, if you remember. And now this double strand is further folded and folded and folded so that it can fit in the nuclear membrane, in the nucleus. So when the cell is not dividing, the DNA, we say it's loosely coiled. That means it spreads out through the nucleus. And we call that the chromatin stage of the DNA. When the cell is dividing, then now the DNA becomes tightly coiled, it's tightly packed, and you can see the chromosomes. So here's an image of the DNA packed in a cell that is dividing. And here's what the chromosome would look like. So the first unit of packing of the DNA is known as the nucleosome. So here the DNA, the double strand, is represented in red. And the first packing or folding goes around these 8 proteins. These spheres represent proteins that are known as histones. So you see we have 8, 1, 2, 3, 4, 5, 6, 7, 8. So this group of 8 proteins with the DNA folded around them is known as the nucleosome. So that's the first unit of DNA packing. It's the nucleosome, and you see here another histone that keeps the DNA in a certain folding position. So, yes?
Professor, what are those ball-like things, like the balls?
Yes, those are proteins. So the DNA is going around those 8 proteins, is folding, going around. So these 8 balls or spheres and this green structure, they're all proteins known as histones, and they are used for DNA packing. So the red represents the DNA, the gray represents proteins that are used for the DNA to fold around them, and so is this green. So this unit with the 8 histones plus this other histone and the DNA is known as a nucleosome. What's the difference between a nucleoli and nucleosome? The nucleoli are these bodies that are located in the nucleus, and they are the sites of ribosome assembly. The nucleosome refers only to DNA and the DNA folding, and it's the first unit of nuclear DNA folding. And so here you see the double strand. Here you see the DNA going around nucleosomes, and then it continues to fold and fold. And fold, giving rise to the nucleosome. So the DNA is 2 meters or 6 feet long. And when it's completely folded, it becomes 50,000 times shorter. So you go from a 6-foot molecule that is 50,000 times shorter to fit in the nucleus. Okay, so now let's go from protein, how are proteins synthesizes. So the first step is transcription. So in the chromosome, you have many genes. So these chromosomes, of course, in our body, they are not colored. This is a computer interpretation. So we have 23 pairs in somatic cells, and each fragment, each section of a chromosome is a gene. So the DNA gives rise to a protein by a process that's known, 2 steps, transcription and translation, with a step in between. So in our genome, the genome is the collection of all genes. So the human genome is around 20,000 to 25,000 genes. So it doesn't contain that many genes compared to, for example, the corn. The corn, the plant corn, has double the amount of genes than we have. And you might say, but how can that be? Or let's say a fly, the fruit fly that you see around the bananas, they have 13,000 or 14,000 genes. How can that be if we are such a complex organism? So in humans, what happens is that one gene can encode for more than one protein. So that's the secret. Okay, so all those 6 feet of DNA, that huge molecule, there's less than 1.5% encodes for proteins. What does that mean? That means that the 25,000 genes only make up 1.5% of the human genome, of the human DNA. All the rest represent regulatory sequences, regulatory molecules that regulate the expression of those human genes. So we say that the rest, the 98.5% of the DNA is non-coding, and it plays a regulatory role. That means that those genes are not going to end up as encoding for proteins. So protein synthesis has these 3 steps. Transcription, where the DNA encodes for a pre-messenger RNA. Then the pre-messenger RNA, as its name indicates, pre-messenger RNA, has to mature. So pre-messenger RNA processing converts the pre-mRNA into mature RNA. And then the first step is translation, where the messenger RNA is converted to protein, and this happens in the ribosome. So you go from DNA to messenger RNA to protein. So the first step, the messenger RNA or the pre-messenger RNA occurs in the nucleus, the transcription step. So only one strand of DNA is used to make the messenger RNA. So the transcription starts at a specific area in the DNA that is known as the promoter. And this promoter is highly regulated because we have an enzyme that binds to the promoter and starts the process of transcription. So you see here the enzyme that is involved in the synthesis of pre-messenger RNA is known as RNA polymerase. Polymerase makes a polymer of the units that make up the messenger RNA. Now, this enzyme has 2 functions. One is to break apart the hydrogen bonds that link the 2 DNA strands. And the next function is to synthesize the pre-messenger RNA. So 2 different functions. And here, the pre-messenger RNA, the units, the nucleotides, are linked by phosphodiester bonds. So it has nothing to do with the peptide bond. Remember, the peptide bond is made in the ribosome and links two amino acids. Here, in the messenger RNA, pre-messenger RNA, you're linking two nucleotides by making a phosphodiester bond. So it's not a peptide bond, it's a phosphodiester. Yes?
Professor, so the DNA polymerase first it breaks, for example, the bond between adenine and thymine, it's gonna break that. And then
Yes.
it's gonna again connect adenine and thymine. It's gonna
Yes.
break and then connect.
Yes.
Okay, so the second time when it's connecting, what is it creating again?
What is what? It forms a phosphodiester bond.
Thank you.
So that links the nucleotides that make up the messenger RNA. So you see here the movement. You see, this is one nucleotide, another nucleotide, another nucleotide. So it forms the messenger, the pre-messenger RNA. As it moves down the DNA, so only one strand is used to make the messenger RNA. And if you remember what is a nucleotide is made up of a sugar, in the case of the messenger RNA, it's ribose. Then you have a base and you have a phosphate group. So these units, these nucleotides, are linked to each other by the enzyme, by the polymerase, RNA polymerase, through a phosphodiester bond. Okay, so let's say you have this DNA with these bases. So what would be the sequence of the messenger RNA? So it has to be complementary to these bases. So the messenger RNA, G would be C, C would be G, and so on and so forth. Thymine would be adenine. Now look here, adenine should be linked to a thymine, but there's no thymine in the messenger RNA. That would be uracil. So remember, if I put something like a question that I give a sequence of the bases on a DNA molecule and ask what would be the sequence of the messenger RNA, remember that there's no thymine in the messenger RNA. Instead of a thymine, there's uracil. Okay, so how is the pre-messenger RNA processed? So the pre-messenger RNA has sections. Some are called exons and some are called introns. So what happens in the pre-messenger RNA process, the introns are removed by enzyme complex known as the spliceosome. So the mature mRNA is made up of exons only. The introns are removed. So the enzyme complex that removes the introns is known as the spliceosome. So you see here these small ribonucleoproteins that make up the spliceosome. And you see here the pre-messenger RNA, the introns are removed by the spliceosome. So what mature RNA, how do you define a mature RNA? It's made up only of exons. The introns are removed. So now let's finish with the translation. What happens in the translation?
I have a question.
Yes.
So, if only one of the strands are used to,
Make the messenger RNA.
yeah, make the messenger RNA, what's the other strand doing?
Well, the other strand stays there, doesn't do anything to make the messenger RNA. Just one strand is used.
So, what happens to it in the end? Like,
It's just the double strand is reassembled.
so instead of so both of them are making mRNA at the same time?
Well, no, only one makes, because that would be impossible for the enzyme to work on the two strands. So the enzyme works on just one strand.
So when the strand that's making the mRNA ends up like, forming the mRNA and then forming the protein, that other strand,
The DNA strand
yeah.
reassembles.
Oh, so it goes back to normal.
Yes.
Oh, okay.
Okay, so now let's go to the translation, which occurs in the cytoplasm. So there are three types of RNA. We already talked a lot about the messenger RNA, and I talked a little bit about the transfer RNA. The transfer RNA is very, very important for translation, and it's located in the cytoplasm. And then you have this other RNA, ribosomal RNA, that's made in the nucleolus. All of these three RNAs are made, all produced in the nucleus. Okay, so let's look at the messenger RNA. So the messenger RNA has nucleotides that are attached to each other. And if you think about three nucleotides, these nucleotides, one has guanine, cytosine, adenine. So these three groups of bases make up what we call a codon. So this codon, so these three bases, this sequence, G, C, A, is specific for this amino acid, alanine. So this is the trick for making a protein that is so huge, and it's based on an alphabet of 4 letters, let's say, because you only have 4 bases, but you get very, very different proteins that are made based on these 4 bases, but you can get a huge variety of proteins. So it all depends on the sequence of these codons. So what I want to transmit to you is that the codon is made by 3 different nucleotides, or it can be the same nucleotides. And so each of the nucleotides has a base, right? So this nucleotide is G, this has the base C, this has the base A, so guanine, cytosine, adenine, and this sequence of this codon is specific for alanine. So here you see a messenger RNA, and you see that it's grouped into 3 bases at a time. So each 3 bases is what we call a codon. So this sequence, AUG, is specific for methionine. And interestingly enough, all proteins, most proteins, start with the methionine. So we call this the start codon, because most proteins have methionine at their end terminus. And so now this sequence of base pairs, which is codon 2, GGC, is specific for glycine. Then you have UCC, specific for serine. So you see that the sequence of the codons is very specific for a protein, for an amino acid. And the sequence of the amino acids is what gives specificity to a protein. So you have amino acid 1, 2, 3, 4, 5, 6. So this protein has these amino acids in sequence. And that's what gives the personality to each protein. Now,
Professor,
yes.
to memorize the names and structures and the codons for the amino acids?
No, you don't have to know that. I would never ask you to do that.
So,
So we won't have to remember this slide then.
No.
Oh.
This is just to explain how each codon that is made up by 3 nucleotides that contain specific base pairs gives rise to or is specific for an amino acid.
So just to clarify, so after the splicing occurs, where like the introns disappear and the exons like come together.
Yes, just 1 minute. Students that have to leave to go to lab, please do, because this is being recorded. So please ask your question again.
Yeah, so after the splicing occurs, where the introns like are removed and the exons combine, that's when
Right.
the mRNA starts.
It, yes. So remember, now this process messenger RNA now goes to the ribosome. And the connection of these amino acids happens on the ribosome, remember? So this just shows a table with the different combination of the nucleotides that are specific for each amino acid. You do not have to know this. The reason why I'm showing this is because there's redundancy. What does that mean? That means, for example, the amino acid serine can be encoded by 4 different combinations of nucleotides. So that means redundancy. So if you have this combination or this or this or this, those are specific for serine. So I'm showing this only to give you the concept of redundancy. That means that each amino acid does not have just one codon. Okay, so let me go to the next one.
Professor,
Yes.
I was just wondering, so the amino acids, so the codons code for an amino acid and then that makes the protein?
Yes.
Okay.
Okay. So you'll understand now here. So here comes the transfer RNA. So the transfer RNA is much smaller than a ribosomal RNA. And it's folded. The messenger RNA is all straight. The transfer RNA has some kind of folding. So this is how it looks like. At one end, it's linked to an amino acid. At the other end, it has what we call an anticodon. So that means that these 3 nucleotides that are sticking out of the transfer RNA are complementary to the 3 nucleotides that are on the messenger RNA. So you have here a combination of a codon in the messenger RNA and an anticodon in the transfer RNA. And this is how they come together to bring the amino acids together in the ribosome. So you see here, this amino acid is phenylalanine, and it's attached to the transfer RNA that has an anticodon at the end. So what happens, so remember that in the ribosome, we had these 3 areas. So these areas is where the transfer RNA binds. This is the large unit of the ribosome, and here's the small unit with the ribosome attached to it. So you see here these codons in the messenger RNA, and here you see the transfer RNA. So the transfer RNA comes to the ribosome, brings a specific amino acid, phenylalanine, and the anticodon is going to be complementary to the codon in this A area. So the A area in the ribosome is the acceptor, where the transfer RNA binds first. And then the P area is known as the peptidyl. This is where the peptide bond is formed. And then the E area is where the transfer RNA leaves once the amino acid is linked to a previous amino acid. So you have a transfer RNA coming to the ribosome, moving to the peptidyl site where the peptide bond is formed between methionine and tryptophan, and then it moves on to go and fish for another amino acid. So this is what happens in translation. So in the ribosome, you have this movement of the transfer RNA with a specific amino acid coming to the A site, moving to the P site, where you have the peptide bond formed, and then leaving out because free of the amino acid. And this is how translation happens. Are the codons connected by peptide bonds? No. The codons here, codons is a term used by the messenger RNA. They are connected by a phosphodiester bond. Are the best examples of questions? Okay, I'm going to post examples of questions today, okay? So the translation involves 3 steps. The initiation, which is the start codon, which is methionine, then elongation, where the peptide bond is being formed and the amino acid chain is being elongated, and then termination, where there's a stop codon that says stop. no more amino acids. And this is what leads to the protein being made. And I'm going to stop here. So remember, you have messenger RNA being transcribed in the nucleus, giving rise to the pre-messenger RNA, where it is maturing within the nucleus. Then the mature messenger RNA leaves the nucleus, goes to the ribosome, to the small subunit, and then the large subunit is a subunit that makes the protein, the polypeptide chain, based on the transfer RNA, bringing specific amino acids. Okay, so any questions? And this is just more views of this process.
I think there's some questions in the chat.
Yes, but I answered all of them. I have labs. Oh, I answered. Okay, Professor. Let's see. Oh, let's see. Professor, for the exam next week, Thursday, do we just study the lecture slides? Then you have gone so far in the last 3 weeks. So for the exam, I'm also going to post what is necessary for the exam. So all lecture slides, all lecture notes, all the recordings, that's all you need to focus on. But I highly recommend that you read the book to understand the concepts that I go over in the lecture. So, Professor, will we expect to learn names and structures of the most common amino acids? No, no way. Professor, please, oh, I have the lab, I already answered this. Or the codons connected by peptide bonds? No, only amino acids are connected by peptide bonds. The codons are connected by phosphodiester bonds. Are the best examples of questions we made on the exam, lecture, practice questions. I'm going to post some questions. There are 60 questions, multiple choice questions in the exam. Do we have to memorize all the material covered from week 1? Yes, all that material has to be known. You have to know all that material starting in lecture 1. Yes, it's all multiple choice questions. So what are the codons of the messenger RNA? So let me go back. So this is one codon. So you think of this, each of these is a nucleotide with the base, uracil, cytosine, cytosine. So this sequence, UCC, is a codon specific for serine. What are the codons? So the what are the codons and anticodons linked to? They're really, this is a complementary linkage. There's really no linkage, no covalent bond here. Because remember, the transfer RNA is going to be moved. So I will tell you how many questions for each chapter. I'm going to put that information today. It's going to be chemistry, it's going to be cell, and it's going to be integumentary system, which we will cover next week. On the slide notes, you give examples of homeostasis. You don't need to memorize the examples, but I'll give you an example of I'll give you examples of loops, and you have to say if it's negative, positive, and so on. Negative and positive feedback. I'll give you examples. You don't have to memorize. What chapters of the book is the exam focused on? 1, 2, 3, and 5. This is all on the syllabus. Are we going to take the exam on paper, or you're going to take the exam on Examplifier in person in the assembly?
Professor, do we need to buy scantrons for the exam?
No, but there's no need for scantrons. The exam is going to be taken with your computers, but in person. You're not going to need scan drawings. Are there any questions on specific examples, clinical applications, or concepts? Yes. Anything that's on the lecture notes, slides, or recordings, you have to know all that. Professor, this is related to this. Okay. For the exam next week, do we just study the, oh, I think I answered this already. What did she say to this again? Study everything, know the slides, notes, recordings. I could not attend last offline class because of moving. So I posted the recording from September 11th. Yes, you need to know disease examples, what is causing these diseases. Yes, you will get the results of the exam right away. If we don't have, Professor, if we don't have during the exam, is it possible to upload the exam answers when I get home? You will be able, I will make the exam available later. I'm going to email you about getting an invitation to create a username. Okay, please do. Please send me an email if you have not yet downloaded ExamSoft. The exam is 1 hour and 15 minutes. If you arrive on time, if you are 20 minutes late, then your exam time will be much shorter. We cannot stay in the auditorium past 5:15. There's a class right over us, so please don't be late. This exam will take 1 hour, 15 minutes to complete? Yes. Next Thursday is, no, this Thursday, the exam is on September 25. But look in the syllabus, we have the syllabus posted on our website. It says very clearly that exam number 1 is on September 25. 60 questions only, multiple choice, not writing, correct. For exam questions on homeostasis, would there be questions where we have identified what the receptor is, the control center? Yes, I will give you an example and you will have to identify what is the control center and so on. Yes. Can we leave after we are done? Yes. Next week, exam 9:25. Yes. Oh, I have to think about this. Okay.
Professor,
Yes.
so the questions that you're going to post today, only those questions will like come on the exam, or like you will ask us other questions.
No, those questions that I'm going to post will not be on the exam. The exam will contain different questions than the ones I will post.
Oh, so this is just for like our concept and practice, right?
Yes, exactly.
So if we study these questions, it's going to help us figure out the actual questions that will be asked on the exam.
I'm going to put examples of questions. What you have to study is the lecture notes, the lecture slides and the recordings, and my lectures, of course. Anything that's there, I can ask, okay? You have to know. So, is there an exam review? So actually, I was thinking of having a review before the exam. So on September 24, at 5:30 p.m., I was thinking of having a review.
I think Hunter followed this close.
That's right. So we'll do it on Zoom, right? So
Oh,
that's
right. So Wednesday, Zoom.
what?
So it's going to be on a Wednesday, right? The Zoom.
Yes.
Okay.
I'm going to do it. I'll send an email. I'm going to do it the day before the exam. So September 24, which is a Wednesday.
Professor, is there any way we can have the review before that so that, we have more time to get an idea, like over the weekend?
Okay, I can do it September 23rd then. No, September 23rd. I'll see. But I think most likely on the 24th. Because like this, you have studied, so you will know more. What questions do you have? You have to study all the material, what is in the notes, the slides, and the recordings. The exam is for 1 hour and 15 minutes. What do you recommend for studying? Yes, you have to study all the material. I don't like the word memorizing because you also have to understand it. But yes, you have to know everything that's in the lecture notes, slides, and recording. I have emailed you about the invitation. Okay, good. I will take a look at the email. Can I install exam soft on both my laptop and no, an iPad? No. You can only install on one smart device. Only one is allowed. Okay? You cannot move from one to the other. Okay.
Professor, like unfortunately, if my laptop like stops working during the exam, what will happen? Like,
You have
will
to
they?
make sure that it works. And you have to make sure that it's fully charged. Okay? Why would it stop during the exam? You know, you don't need Wi-Fi when you're taking the exam. So I don't see a reason why your computer would stop working. You don't need Wi-Fi when you're taking the exam. The exam will be automatically downloaded onto your computer without you requiring Wi-Fi. Okay? So, right. So if I say I post the review, then who's going to show up? One student, because they think, oh, she's going to post the review. I don't need to attend the review, right? So that's the thing. If I don't record the review, this will make students come in. If I post a review, then no one will come in because they think, oh, that I'm going to post a review. The exam is going to be available to download 48 hours before the exam, so on the 23rd. But you won't know that it's there. You won't know where it is. Okay? Okay, anything else? What time is review going to be? It's going to be on Wednesday at 5:30. Okay? So are we good? All questions asked? Any more questions? No, the review is not tomorrow because the exam is on September 25. So next week. So the review is going to be on the 24th of September. Okay? No, we're going to use the same Zoom link, not a different one. I don't know. All these students want to come in. No. Is there a separate? What if you have lab at 6 p.m.? That Wednesday, there's no school. So nobody has class on the 24th of September. There's no class. That's why I chose to make it that Wednesday. There are no classes. Okay? I know people have jobs, so that's why I'm doing it at 5:30. So okay. Any more questions? Okay. Seems like we're all done. Good night, everybody. Bye-bye. See you Thursday.